We're ready to resume. Oh, and okay, Mitch is going to lead the way in gathering questions and distributing. Thank you.
Okay. All right, so I'm pretty much going to pass on all the questions to my colleagues since I am admittedly a clinical physician, but this one's probably best for Vik. This is more basic science. So this question is there any known way to stimulate the dormant brain cell? And a follow-up is the TMS device, will that work, or will DBS work to stimulate dormant brain cells?
So I guess the reference there to DBS makes me think by stimulate, the question means electrically activate. That's what DBS and deep brain stimulation or transcranial magnetic stimulation are ways of activating brain cells. I think that you know that that's been very successful for Parkinson's disease, and one of the reasons there is that the circuitry in Parkinson's typically is well defined, and that you can substitute in some ways for that lack of dopamine by using stimulation in some ways to bypass the effects of dopamine. In MSA, there are the circuitry that's affected is more complicated and broad, and so it is harder to conceive of a treatment, a discrete treatment, whether that's replacing dopamine cells or using deep brain stimulation. It's harder to conceive of a discrete, one kind of shot treatment that would help in MSA. But I think that there are different ways of stimulating cells or nurturing them, and that's what I think Daniel was referring to with some of the treatments he was thinking about or discussing, like mesenchymal stem cells that are meant to increase growth factors that can help broad parts of the brain. And so I would guess that with our current state of knowledge, we would think that broader therapies that affect different parts of the brain at the same time, whether that's through growth factors or through affecting this protein synuclein, might show more promise than therapies that really target one type of the brain, like electrical stimulation or deep brain stimulation.
So, good questions. As I expected, I told you this is going to happen. So good questions about, someone's been reading about hemochromatosis, it seems, which is obviously a disorder where the body doesn't do a good job with metabolizing iron. And the insightful question is, are there neurologic changes of hemochromatosis that are similar to the things we're talking about? Is there a relationship between hemochromatosis and MSA? I would say, I have personally not seen any hemochromatosis patients develop MSA. There are reports in the literature of people that have movement-related symptoms like extrapyramidal syndrome that are been described with hemochromatosis, and then there are other disorders where you can see iron accumulation, where patients can have movement problems. I haven't seen it primarily with MSA though, but I think it's an insightful connection that there are diseases that do affect iron and they do have brain-related changes. They're just not identical to MSA, and that certainly is a link. And then following up on those, can we measure serum iron with brain iron? Is that a relationship? Probably not. There's probably not a clear direct relationship between serum and brain iron. And then what's the downside of iron chelators? And I assume that there are questions about ways to basically pull the iron out using chelation therapy. So there is a study in Europe, I believe, looking at iron chelation in Parkinson's disease, and that's ongoing. Chelation therapy is not without safety risks. There are some things that one can happen, so we generally don't recommend it unless there's a real strong medical need for chelation therapy. And certainly there's an idea that if you chelate something peripherally, does that affect the central, i.e. the brain? And that's certainly not entirely evident, which is why we're looking, why small molecules that can cross the blood-brain barrier and change iron efflux and things like that are attractive from a drug development standpoint. And then a side note is downside of starting CoQ10. I don't see any clear clinical downsides to starting it aside from it costs money and it doesn't have any clinically proven efficacy. Some of my patients ask to do it and I say it's fine, but I don't. There's no clear side effects in my clinical experience with using it, unless my colleagues have something different to say.
I guess the only thing to say with that is typically people use it at quite high doses based on the literature from mitochondrial diseases. And so typically people are taking it around 1,200 milligrams a day in divided doses. And if you were to buy that at the pharmacy, that would be extremely expensive because most of the time it's 100 milligram tablets. Getting it on the internet from a reputable source is definitely a cheaper way to do it. But if you're going to do it, you might as well do it at the dose that has been shown to be effective for other diseases like mitochondrial diseases.
We have a follow-up question on CoQ10. Would taking supplemental CoQ prevent being included in a clinical trial?
So my answer to that would be I don't think so. I would say no, unless it's clearly an exclusion criteria, because CoQ10 has no clear proven efficacy to modulate these diseases. I don't think it'd be an exclusion. Okay. Unless you're Japanese.
Yeah, I think it would just be if the particular trial was actually studying the biology of something related to CoQ, then it might be, but I wouldn't think it would be classified as a supplement, so typically not.
And we can go down the line. I'll take this one. So specifically, since the question is Stanford participating in a study? The answer is yes, we're participating in the natural history study, which is the observational study, but we don't have any treatment studies that we're currently participating in. All studies that are open to enrollment, you should be able to find on the website, clinicaltrials.gov, and you can search by MSA and by site, and there should be a contact. My email is on there for the natural history study. If you have any questions, you can feel free to email me through that. And then this is also related to that study. Is there a fee to participate in the observational study? We discussed, for example, if you're a Kaiser patient, which will not pay for testing at non-Kaiser facilities. So, the study doesn't. The funding is always a challenge. So the study doesn't really pay for the patient to have the testing. The funding that's supplied is not enough to cover all the autonomic testing. But as I mentioned, it is kind of standard. It is standard of care. So really, if patients are having autonomic symptoms, they should be having the testing. And Kaiser will pay for the testing and for a consult with us because Kaiser doesn't have autonomic testing capabilities. So that's being a Kaiser patient wouldn't exclude you from the study. You would just have to ask your Kaiser doctor, and then they would authorize the visits, and it'd all be covered through Kaiser, and you could still be enrolled in the study.
I want to ask a follow-up question. How many people use clinicaltrials.gov as a resource? How many people find it to be very cumbersome? Yeah. So I think we got to do it, right? We got to come up with the MSA clinical trials. So that's something we need to do. Don't you think? Let's do it. Yeah.
Absolutely, and I mean, I'll second that. In the GLOMSA, yesterday we had a session on GLOMSA, and that very question was brought up, that it's cumbersome. Patients are going in, they're lost. So if we all have, we've heard that multiple times, and we have to do a better job in getting the information into your hands in a digestible way that you can understand it, right?
Lucy, do you want to take this question here?
The question I got was similar to the question that Mitch got about clinical trial participation and trials that require autonomic testing, and if there are funds and resources to pay for it. And I think here, when we're talking about clinical trial testing, is an important difference is whether this is an observational study that's collecting, your standard of care clinical information like we are doing in the natural history, or if it's an interventional study. Now, if it's an interventional study and you're taking a compound that we think may help change the progression of the disease or improve your symptoms, you shouldn't be paying for that, right? I mean, that's a clear definition. When it comes to autonomic testing for an observational study, I mean, different sites have different rules. And the investigators team that we have, the 17 sites or 20 sites soon we'll have, are all really committed. So if you want to participate and you're close to a recruiting site, have that conversation with the coordinator, find the name of the person, call up and say, look, I really want to be in this study. I know how valuable it is, because different sites have different rules. So, really be proactive for yourself and share with your physician that this is something I really want to do.
We have a trial on chemotherapy. What can you tell us about trials involving chemotherapy? Our movement disorder specialist mentioned there are studies going on that we don't know, and we don't know more than that. So I'm not sure what this meant by the chemotherapy, but
Yeah, nilotinib.
They're talking about the nilotinib. Yeah.
You want to take that?
Parkinson's. Yeah, I'm
There's two of those, right?
Yeah, but not for MSA.
No.
Yeah, I mean, I think these are ongoing trials. I'm guessing the reference here is to a drug called nilotinib that showed some really interesting, striking findings in a very small group of patients. And so I think the trials that are ongoing are rational, well designed, and they're taking this into the appropriate format now. I don't think we can conclude anything from the previous work, and that's exactly why we need these trials, and they're ongoing, and we should wait. And then there is always the possibility that mechanistically this drug is working in a way that might help other synuclein-related diseases, and so it's of interest to MSA patients. But when the mechanism of protection is not clear, and this is getting back to the needs of basic research, it's very difficult to advance a therapy or understand which other diseases it might be relevant for. But again, in that category that I suggested, really good observations from physicians has been very important for neurologic diseases. If this was an important observation that ends up impacting patients, then that's great. But at this point, I would say we don't understand the mechanism, and we need more definitive clinical trial data.
okay. So this is a very good question. I'll open up to the whole panel. Wouldn't a prion-like behavior of alpha-synuclein reduce the likelihood of positive outcomes for stem cell injections in the study shared earlier due to seeding?
I'm telling you, these are like really hard questions.
Yeah, that's definitely too hard for Daniel, so I'll take it.
I'm from Tennessee, man. It's like simple.
I wasn't going to mention that, but since you have,
Your accent's better than mine, though.
I'm way down South Australia. It's even. So I think the question relates to whether the mesenchymal stem cell treatments that are being tried and are very interesting would be affected by this prion-like spreading. The answer there is no, because the mesenchymal stem cells are just being injected to essentially release, this is the presumed mechanism, release growth factors that are helping the brain cells cope with the toxic protein. And they're being injected, at a distant site. So typically we wouldn't think of these types of stem cell therapies as being affected through this prion-like spread mechanism, even if that's what's occurring in the patients. The prion-like spread mechanism may be more relevant when you're transplanting cells into the brain that are actually, meant to be forming connections. Like in the case of Parkinson's disease, you're putting dopamine neurons back into the brain. They're right in the vicinity of where that synuclein is accumulating. So there you might think that this kind of spread would be a problem, but not really for the mesenchymal stem cells that are not being put in there to build the circuitry of the brain, but rather to secrete nutrient factors for the brain. So the mechanism is different.
And to follow up on that topic, one of the questions was, why does it take so long to get to where we are? And I would emphasize that, these trials take a long time to design, to do the proper safety studies, and I agree with you, it is frustrating, but I do think that we're at a place now in terms of stem cell trials that I think we will hopefully see some more studies come in the future. One interesting question about the stem cell trial also was, how do you handle control groups? So if you have an intervention that you're injecting, how do you make a control group? And so usually what you do is you just do the same intervention, but you blind the investigator and the patient to whether or not they're getting a stem cell or just saline. And so usually you have a control in that regard. And so that's one way to handle it.
So the next question is, does participation in one clinical trial preclude you from any other trials? So the answer is not necessarily, but again, that's where those exclusion criteria that Dan mentioned are important. Especially, the observational trials, almost never. You should be able to participate in as many of those as you want. The treatment trials, if you are getting a drug, of course, then that may, make it, exclude you from participating in other treatment trials because, the investigators wouldn't know which drug is working if they're blinded to it.
And I think to echo that point, if you are in a treatment trial and you're in an observational study at the same time, be open. Tell the person collecting your information in the observational study, I'm participating in another study, because we can log that information, and then when we get the data on the back end, we can say, okay, this patient was treated or participated in this particular clinical trial, because all of that information is useful. So the more information you give us, the more opportunity we have to look at different subgroups, different interventions, and those things. So it doesn't preclude you in observational studies.
I might just speak to something that Daniel just mentioned. We have brought this up in previous years, but there is this concept of stem cell tourism, getting stem cell therapies in places where the protocols are not really established, they're not part of a clinical trial, there's no published data. And these trials, as much as I think we all here fully recognize why someone would be interested in bypassing the typical clinical trial mechanisms, because they are, cumbersome and they do take a long time. We understand that, but it's also really important not to enroll yourself in something or be involved in something where you're going to end up more sick than you were before you took it. So as much as that's hard advice to give sometimes, doing the trials a proper way, and I think the Mayo Clinic right now is doing the mesenchymal stem cell trial the proper way, I think it's really, really important.
Okay, is eating high-protein diet good for MSA or not? I don't know if this is a research question, but I don't know if there's any research on protein.
You talk about protein and levodopa.
Yep. So we know that, for instance, in Parkinson's patients, patients taking levodopa, protein can inhibit the, absorption of the drug. So if you're taking certain medications, it can possibly slow or inhibit absorption of medication, which we don't think as much about in MSA. From a clinical perspective, like what we talked about this morning, protein is less offensive than, say, carbohydrates for dropping your blood pressure, so we really don't have a big problem with a high-protein diet. Does anyone have any other comments on that? Okay. And then the next question, is it true that Lyme disease symptoms mimic Parkinson's and MSA? What is your opinion on that? Can I just say no comment, or? The neurological Lyme disease is very rare. It can occur, and Lyme disease is one of those conditions that can cause almost any symptom. But especially on the West Coast, Lyme disease is becoming more of a problem, but it is still very rare. Almost always there are some other signs of that there's been a tick bite with a rash and that you've been exposed. And if the testing for Lyme is negative by the Western blot and the FDA-approved testing, we believe it's negative. There's several companies, including one called IGeneX, which you'll get many more positives, but some of those companies aren't really regulated, so it's hard to interpret the data, and we certainly wouldn't want any patient to get harmful IV antibiotics and things like that if it's not a clear diagnosis. So it's very rare, we'll say. It's just, very rare.
So in New England, where I practice, where Lyme disease is quite common, there are a lot of places that that offer long-term intravenous antibiotic therapy and other things for patients who have suspected Lyme, and this has been quite detrimental to some of our patients because they often are on these kinds of therapies for a long period of time without any with potential toxicities, but also not getting the actual medical care that they need for their diagnosis. And so once again, it's worth being a little careful. We all want therapies and we all want, I understand that, but we certainly have seen patients go to some of these clinics, and I don't think it's been very helpful. So it's good to be cautious.
I just want to make a plug for nutritionists. So I have a couple patients in the last 3 months that have really benefited from a nutritionist, going back to that protein diet question. And if you find that you're losing weight or you're confused about what you should or shouldn't be eating, nutritionists are worth the $60. That's usually what they bill. They pay like $60 for a visit. But I've had a couple of patients in particular that have been really found it beneficial, and it's something that we're trying to put into our clinic, but Medicare doesn't reimburse for it unless you have kidney disease, which is a whole other matter. But I do find that nutritionists can be very helpful if you're trying to navigate food, especially if you're having swallowing problems, because that can happen. So I just want to put that plug in.
Okay, we have a toxins question. I feel strongly that environmental toxins play a big role in MSA and other neurological disorders. I worked in a shipyard prior to going into law enforcement, and several coworkers have Parkinson's. Has there been a study tying MSA or Parkinson's to toxins?
Well, you go ahead. Yeah. I mean, it's certainly been looked at, and there are some toxins that may have relationships to Parkinson's, like rotenone, which is a pesticide that's been studied. It's argued. There's a lot of disagreements about it in the field, I think, but I think it's certainly plausible that you could imagine the combinations of your genes and what you're exposed to could manifest with some kind of pathology. But the actual toxin itself, I think in my interpretation of the data, seems to be still unclear. I don't know if you have a different opinion, but...
No, I think that's right. I mean, it kind of speaks to the point that we need the quantitative data I was mentioning when I was talking, and I think for Parkinson's disease that quantitative data is emerging. So there's two issues with environmental exposures. Do you have the evidence that you need based on the human population exposure data to actually say that there's an association? So that needs really good what's called quantitative epidemiology, and needs numbers and careful studies. And so for Parkinson's, I mentioned Dr. Ritz's work at UCLA. I think that's been really elegant work that has quantitatively shown that certain pesticides are associated with increase of Parkinson's risk. And the second aspect of environmental studies is, even if you point out that there is an association, you do really need to come up with a mechanism and figure out how is this actually happening. And so in one example that I was involved in, where I think you can start to see the reasons for doing different types of research, epidemiologists had pointed out that paraquat increased the risk of Parkinson's, and some years ago when we were studying in stem cells from patients with Parkinson's the biology of how alpha-synuclein is disturbing the biology in patient brain cells, we noted that the way in which synuclein does this is very related to a particular toxic mechanism of paraquat. So there's a couple of different groups working together, but suddenly then something moves from being just, an observation in the field to having some kind of mechanistic basis in the lab. And I think that's what you need to really start to shore up your evidence for an environmental factor. And so for MSA, we can piggyback off the Parkinson's field, assuming some of these things are related. But we are going to need better data to even begin the process in the lab. And so we get this question a lot, but we just don't have the richness in the data sets yet. I think this speaks to a question that in some way to what I have here. Is there genetic or other information that family members should be donating that would be useful for research? And I just want to underscore the importance of what my colleagues here on the panel are doing with the natural history study and with GLOMSAR, because yes, we do want you to submit this kind of data, whether it's genetic factors, family history, or environmental exposures, but we need it submitted in a way that's organized and coherent and that actually adds up to something. Because if people are just giving this information randomly and it's not being collected in a systematic way, we're not going to be able to do anything with that. So this is a partnership between all of us and you to create systems where you can submit data and information that is going to be critical, but submit it in the correct way.
And this is another question that relates to the observational study, the natural history study, which says, do previous studies on autonomic function tests suffice for the observational study? And I would say yes, because in the natural history study, you have the ability to provide what we call retrospective data that was collected at other sites by other physicians, and you can take that information to a site, and that site can use that information. But it's not just autonomic function tests, things like polysomnography tests. If you had a sleep study and in the past, no matter how long ago, and it showed that you had REM sleep behavior disorder or had pauses in your breathing when you were sleeping, take that information, because all of it is useful to us. Some sites will require and will repeat the autonomic testing data, because what we're doing in the study is we're doing standardized autonomic function tests. So that means all of our sites are doing exactly the same autonomic function tests, so that the information we get can be collectively done and pulled together, and there's less variability in it. So please, when you go and you are participating in this study, take your records. Say I had a sleep study, I had a tilt table test, or whatever else you think is relevant, because to us it's really important information.
Was there a time frame for when the studies would be valid? I thought it was 1 year.
We wouldn't necessarily have to. If it shows neurogenic orthostatic hypotension, which is it's a problem with the nerves that doesn't go away, right? So if it shows NOH, it may not necessarily need to be repeated in 1 year. If the autonomic function tests are previously further back, we would like to take that information of what it said at that time and then repeat it again if it was too far out.
We're getting through these. We have lists from the online questions, but I think most of these have been answered. Has research shown links to environmental causes, which Dr. Khurana touched on? Is MSA hereditary, which I think we touched on? This is an interesting, maybe partially touched on, but we can discuss this. Is MSA equally distributed geographically, or are there clusters? Or do we know?
I don't think we have enough numbers. It's rare. Great question. I'm not sure. Well, we don't. I think it's a rare disease, so we don't have a really good sense of clear numbers on whether or not there's certain areas that have more or less. That'd be my answer.
Which country can we go to for stem cell therapy at this moment? Someone must have just walked in, I think.
They didn't understand. You want to go through it again? Silent.
None.
Yeah, I would say be cautious about that. it's not going to be too long before we have good definitive data on this. So as hard as that is, I would hold out to wait to see what happens with that study. The one, I mean, Daniel can correct me if I'm wrong, but my understanding of the early mesenchymal stem cell work at the Mayo Clinic was that the controls, so Daniel talked about experimentals and controls, the controls were historic, I believe, and that's a really important point. So some of the patients got the mesenchymal stem cells in that, but they were compared to patients from before who didn't receive that. So it's really important for the Mayo Clinic to follow up this study with, a study with an actual control group that's getting a sham operation, getting the same procedure but without the stem cells, so that we can actually figure out, was it the stem cells or was it something else? So again, I'm not as familiar with that, but I think I'm saying the right thing there. And so that's why a little bit of patience there is important. I think the Mayo people are doing a great job and doing this as quickly as they can, but we need to see what happens.
So a follow-up stem cell question. When is the expected date to start phase 2 of the, I assume, the Mayo trial?
I think I don't know, and I think they're trying to establish funding, I think is the clear thing. So the only point I'd make about it is, being in a clinical trial is not the same as getting a therapy for your illness. And that's a really hard thing to fathom because, like some of the trials, people think that if I get in the trial, then I'm going to be cured, right? Or I'm going to have a chance to be cured. And I think a lot of times what I spend time counseling with patients is being in the trial may help the next generation more than it may help you. You're committing to being involved in research that will carry the field forward, but it's not a replacement for treatment. And that's a really difficult concept because I appreciate how desperate you are when you have a disease that doesn't have a cure and you have an option that may help you, and you conflate the two to be the same thing. And so I do want to make that point because I find that as a clinician, I spend a lot of time talking to my patients about the difference between being in trial and then getting an experimental treatment that will have being in a trial and having a cure. And so I think that's an important thing to wrestle with and to make the distinction between the two. The same goes for the question about stem cell. What's the motivation for you to want to get the stem cell treatment for which country has it? What's motivating that? Is it because you're expecting that the stem cell treatment is going to change your disease course? We don't know that. So, having a clear motivation, I think, is useful.
Has anybody reviewed Korean data? They have a high stroke and death rate, but they also report those who don't stroke out. Certainly. Has anybody reviewed the Korean data as to what they mean by improvement? They do report a high stroke and death rate, but they say that the patients who do not succumb are markedly improved. What do they mean by that? I can't find it.
You want to talk about that? I don't know very well.
So there was a pretty short study, and I think there were a couple of things that should be just mentioned. It was a very different study to the one that the Mayo Clinic is doing because, as Daniel mentioned, the Mayo Clinic study, they're introducing exactly, so this is intrathecal and not into the blood. And so the Korean study showed when you looked at a particular type of MR imaging, you were getting microhemorrhages in the brain, and so that was certainly a worrisome feature, but it was something that was really looked at. It was really identified on imaging. It wasn't big clinical strokes or hemorrhages. It was microhemorrhages that to my knowledge, is what they described. But when they looked at in a short-term trial, when they looked at the clinical progression of those patients, they showed what they considered a slowing. They did a randomized trial, a slowing of the progression of the MSA using some of the clinical scales that we've been talking about. So there was an imaging-related adverse effect, and then there was a clinical improvement. They obviously had to flag the fact that they were seeing microhemorrhages because longer-term treatments could lead to much more significant clinical strokes. So that is how you reconcile those two things. That's my memory of that particular study. If others know more or differently, then please chime in. But that's my recollection of it. But it's a pretty short-term study, and the MRI raised that red flag. And that's one of the reasons the Mayo group, I think, took a different approach.
Did the Mayo group not initially do intracarotid infusions?
All right, and then we're going to have to, sorry.
They didn't do it. Did the Mayo group not do any intracarotid? Okay. Thank you.
All right, we have one more question for Oscar Ogans. He says he has a question that he's been trying to get an answer to since yesterday, and if he's in the audience, we can give you the microphone. Sir Oscar? Okay.
I'm good. I think everybody's going to...
No, you need the mic. Oscar, you need the mic.
Okay, thank you.
Come on up here.
I have one short question. I've heard about people with MSA with the low blood pressure. I want to know, do you have any knowledge of any cases where the blood pressure is extremely high? My wife, for the last week, her blood pressure has been anywhere from 170 on up. Can you tell me about anything? I put in the question since yesterday, and it seems like everybody that got a hold to the card just avoided it or didn't have an answer for it. If you don't have an answer, just say so.
Oscar, I think what you raised here, if I can take this one, it's a really important issue, right? Because we talk a lot about low blood pressure, about orthostatic hypotension. But when patients with MSA are lying flat, the blood pressure can be very high. They have something called supine hypertension, which translates in simple terms to high blood pressure when you're lying down flat. Okay? We, together with the American Autonomic Society and different investigators, we've recognized that supine hypertension is a problem. We've put together guidelines on how it should be recognized in patients with MSA, and we've reviewed what available treatments they are. If your wife has high blood pressure, it's important that you know, and it's important that you have that information, because if she's in the emergency room or the doctor's office or something like that, you should have the knowledge that high blood pressure, supine, is part and parcel of the disease. If you have an autonomic problem and if you sit her up, the blood pressure will get lower because it's lowered by gravity. And if you stand her up and she does have autonomic failure, it will become even lower. Right? So I mean, anybody on this panel will be happy to provide you support for, how to manage supine hypertension, because it can be treated, and there can be things you can do at home that can help with the high blood pressure.
Okay. Because hers is even high when she's, I'm sorry. Hers is even high when she's sitting up. I don't check it when she's laying down. It's always when she's sitting up. And it can be like 179, and I tell her wait for a minute, and check it again in 5 to 10 minutes, and it might drop a little bit. Sometimes it'll drop a lot. But this is like after she wakes up in the morning, she takes some medicine at night that's supposed to lower it. When we first started giving it to her, like right before bedtime, it was fine. Her blood pressure would be normal in the morning, and now the last few days it's been up in the roof. Okay. I thank you very much.
One possibly encouraging fact that, at least from data that we have, is the high blood pressure, while it is potentially dangerous, it rarely does actually lead to very serious problems, stroke and heart attack, because usually there's these big swings, and patients have typically more low pressures than high. So if it's just a few hours throughout 24-hour period, those highs aren't as dangerous or as concerning as the lows, and don't cause a lot of the heart and brain disease that we usually worry about in patients that have high blood pressure all the time. So it's not as life-threatening or as dangerous as people may think.
And just another thing to add to that, Oscar, take the blood pressure reading log. We love when people come to the autonomic clinic with, this is my wife's blood pressure. This is her blood pressure when she's lying down. This is her blood pressure after a meal. This is when she take those numbers, because there's no, you're with her all the time. You see the blood pressure changes, and it's really useful for my colleagues to see those numbers, right? So that's powerful information. Take it. Talk about it.
All right. Thank you very much. Thank you.
Thank you. All right, is that all of our questions? All right, so a very big thank you to our panel, Dr. Lucy Nordcliffe-Kaufman, Dr. Mitch Miglis, Dr. Daniel Claassen, and Dr. Vik Khurana. Thank you so much.